All right, so when we look at what we've done so far with polymers, more than one mer, more than one polymer. It's actually, I shouldn't say more than one. I should say, oh, could you put those in there? Thank you. Sucrose or lactose, there are two examples of disaccharides. With the polysaccharides, I would expect, and I think College Board expects, that you know what cellulose is.
You don't have any more in the back one.
Oh, there are. I don't know where they are. They might be in the back room. Cellulose and their linkage. You should know. I'm sorry, I'm blanking out here. Starch and its linkage. And you should understand glycogen and their linkage. Alpha 4, alpha 1,4, beta 1,6, whatever. The monosaccharides that you should need to know. Definitely you need to know glucose. That's going to be your friend. You certainly should know ribose very well. These two you should know very well. You should know that glucose is a structural isomer of fructose. You should know both of these are hexoses, 6 carbons. You should know ribose is 5 carbons. This is 5 rings, ring structure. This is 6 ring structure. 6 member ring structure, right? Where ribose is 5 and 5, right? 5 carbons and 5 member ring. So the pentagon versus hexagon. I think that should be clear as well. Should you know galactose? Should you know? The other one up here you might see is maltose. Should you know those? Yeah, you can. It was not going to hurt you, but knowing this, I think is definitely going to be a solid. You'll think it'll take you through. All right, so make sure you know this stuff. So fats, lipids. So we finished that. And we're all in agreement that we hit that pretty hard. We're all good with that. No questions, no concerns. Oh, don't forget that all these linkages, they're all connected to what's called glycosidic, meaning there's sugar having to do with sugar, glycosidic bonds. All right. That's that dehydration synthesis. That's the other thing you should know. You should understand anytime you're making these polymers or you're linking these monomers together, and in fact making biomolecules at all, you're dealing with dehydration synthesis. Bless you. Number 1 rule in biology is never say all. Never say all, because you're always going to find some ridiculous exception. So dehydration synthesis, that looks awful. I'm going to erase it. I'm sorry, I'm petty. I can't help it. No, I messed everything up. It just looks ugly. I can't handle it. All right, so through dehydration synthesis, you can take this alcohol, sugars. Remember, sugars, by the way, end in OSE. That's the ending of all sugars. All carbohydrates. This whole thing is called, all these are carbohydrates. Sugars are generally thought of as like monosaccharides and disaccharides. Polysaccharides are generally known as carbohydrates, just generally. It's kind of a rule of thumb. The whole classification is carbohydrates. So when people talk about not eating carbs, they mean sugar and starch, right? Pasta is also carbs, right? So when they say carbs, they mean carbohydrates. Carbohydrates. It's all these different kinds of carbohydrates. Now, with lipids, we did talk about the idea that these include fats and fatty acids and also some hormones known as steroids. Things like cholesterol are all lipids. Tend to be non-polar. They tend to be, and this is a question people are like, isn't there oxygen in lipids? Yes, there's oxygen in lipids. There is. And sometimes there's other elements in lipids as well. Like there's phosphorus in lipids, right? Especially phospholipids. But the key is they're mostly carbon and hydrogen. So they're also known as hydrocarbons. Because they're mostly hydrogen and carbon. Now there are other elements involved, but you saw in all the example questions you went over yesterday, people kept asking me, we walked through it, right? And I posted the video. You saw that some of these hydrocarbons, that they were going to ask you that you need to know what elements are involved in each of these types of biomolecules. So for carbohydrates, it's C, H, and you know what? C, H, and O in a 1:2:1 ratio. Most carbohydrates are going to be one. Are there going to be exceptions? Yes. But in general, the rule is it's a 1:2:1 ratio, right? So you're going to see what is the rule of thumb when you're looking at a molecule. If you see about as much carbon as you do oxygen, then it's not a lipid. Because if you see every time you see a carbon, there's an oxygen. It's not a lipid. Could be just a regular carbohydrate, or it could be an alcohol. Alcohols all end with OL. Glycerol, ethanol, methanol. They all end with OL. It's an ending. OH groups. Alcohols have OH groups, and so therefore, in a way, it gets a little confusing where you draw the line between carbohydrates and alcohols. But alcohols are carbohydrate. I know you've all heard of beer bellies. Beer bellies happen because people drink beer and they think this alcohol is it's better than eating pasta. But no, it's exactly, calorized is exactly the same. You're just drinking your pasta. You're drinking your calories. So it's just another, your body treats it like a carbohydrate.
Okay, so I'm not
All right. So fats, fatty acids, steroids, cholesterol, these things are called hydrocarbons. So the one we talked about was combining alcohols with these long chain lipids. Tend to be these two here, especially this one, are long chains of carbon and hydrogen. When you go to the gas station, anybody here drive? You put gas in the car, anybody fill up a gas tank? Have you seen someone fill up a gas tank? So there's different types of gasoline, right? I think once that I always use the lowest octane. It's called octane. Do you know what octane means? How many carbons? The longer, well, no, it's a good thought. Octane means a good. Fine. The higher your octane, the more carbons you have. So when the number, when you see the number on the gas tank, the longer the chain, because it's a chain of carbons. Gasoline is a lipid. It's an oil. It's made from crude oil that's refined into gasoline. And so we take this long chain of carbon and hydrogen. It's also known as a hydrocarbon, by the way. So longer the chain, should you use the long chain, the biggest number, the lowest number, most cars are going to want the lowest number. It's going to run with less NOx. So it takes less to blow them up. You're blowing them up in your car. That's what you're doing. You blow them up in your engine. That's why I call it internal combustion engine. We blow up the gasoline that pushes those pistons and turns your wheels. Yeah? Polyunsaturated, yeah, you'll need to know that. So is gasoline polyunsaturated? Probably saturated, but I don't know. I've never looked at it. Probably I would say saturated. So gasoline saturated. Well, is it? Is it because it's liquid? It's liquid and it's complicated because gasoline's been processed. This isn't just oil. Gasoline has been a processed fuel, and I'm gonna argue. I don't know. You want me to look it up? We talked about how fats can lay on top of each other and make solid, and why saturated is solid and liquid. The other part I left out was the size. The larger the molecule, the more likely it is to be denser. So more likely it's gonna be solid at room temperature. Thank you. The smaller it is, the more likely it's going to turn to gas. I mean, that makes sense, right? So these are small, saturated, but small. So that's likely the problem. They're not. The other key is obviously polar and nonpolar. If something's polar, it tends to be liquid or solid at room temperature, or something's nonpolar, it tends to be gas at room temperature. So the small nonpolar molecules tend to be gas. So even though it's saturated, it's small, and when we're talking about fats, and let me look at these big long chains of fatty acids will be solid at room temperature because they're saturated, they can lay down, and they're long. So you have this end of the fatty acid when we're looking at fatty acids. You have to know these functional groups because it's going to help you long-term and short-term. But one of the functional groups we talked about is C double bond O, OH. That's that carboxylic acid head. That's at the head of the fatty acid. Then after that, you just turn into a bunch of carbons. And as we saw with gasoline, you're only talking about 10, maybe 12. But some of these are 20 or 30 or 40 carbons long. So they're long chains. They're straight. If they're saturated, they're straight. And this is messy, but I'm just going to go ahead and I'm going to keep it short because I am not going to deal with it. This is not an organic chemistry class, so I don't mind. So I'm not going to sweat it's naming these or what have you, and just say, look, let's just take a look at this one thing, and let's see how we would connect the two of them. If we had two of them, if we had three of them, how would we connect them to a glycerol molecule? And I showed you that already too, right? So we have 3 of them. How do we connect these to? We're obviously going to use dehydration synthesis. We do have three glycerol molecules, 3 carbons. I'm going to exaggerate the bond lengths just to give myself some room. You have OHs. You have H's. This is an alcohol. What do you notice about this alcohol? The ratio of carbons to hydrogens to oxygens. It's 1 to 2 to 1.
Yeah, it's carbon.
Right? It's a little more, right? It's like 1 carbon, 1 oxygen, 1 carbon, 1 oxygen, 1 carbon, 1 oxygen. Everybody agree? And we got 3 of these, and 1, 2, 3, 4, 5, 6, 7, 8. We have not quite, wait, I'm sorry. 1, 2, 3, 4, 5, 6, 7, 8. Yeah, 8, not quite. It's more than double, right? It's more than double the number of hydrogens, but it's 1 carbon, 1 oxygen, 1 to 1. You see the ratio, what I'm talking about when I say ratios? Okay. All right, if we're going to combine these, if we're going to make them into one thing called triglyceride. This is a triglyceride. Triglycerides are very important.
There's another question.
Mm-hmm.
So when you're writing HO, does it matter if you write OH or HO, or does it have to be?
Not really. It depends on how you're aligning the bonds when you're drawing them. These are drawings, so they're sketches. They're not really meant to. Now, HO or OH, there are conventions. You'll learn them when you do organic chemistry, all right, in college, so don't worry about it. If you're drawing it on an AP exam, I can't imagine somebody's going to gig you on it. You might find some weird grader that's going to say, hey, they drew HO instead of OH, that's wrong. They will not mark it wrong on the paper because there's a rubric, and I've never seen that in the rubric. Like, that would be ridiculous. At that point, you're just getting too nitpicky. I think they'll be happy that you got it right. I don't think they're going to be worried if they put HO or OH. They're not going to have you draw it out, though. That's not happening. I can't imagine it. They'll give it to you, and they'll expect you to know what dehydration synthesis is. And they'll ask you to describe it, and drawing it out will help you describe it. So let's be clear about that. So being able to draw it out will help you to sit there and explain. Yeah, like so I can easily see them asking is one of the FRQ questions, little I, how are biomolecules connected? Or explain dehydration synthesis. I could easily see them asking you that. All right. So that's when 2 biomolecules come together. Usually there's a double bond involved because the double bond is an extra space there for you to, but we'll see that in cases where usually the carboxylic acid is important because an acid is willing to release a hydrogen and we're ready to go. So what will happen here is an OH and an H will leave and water will come out. So water will come out and the double bond O stays. So what's going to happen is there's going to be a carbon that's going to have carbon, O, carbon, double bond O sticking off to the side. Does that make sense? So let me just get the picture. I'm going to have to draw it because it won't let me, the internet, I don't know why. It's very frustrating. By the way, YouTube videos have been blocked out again by your computers, but if you go into Teams, you can access educational. So access your YouTube video through Teams on our network. I don't know, a kid did it and he showed me. I didn't know how he did it because I don't need to do that. I can't keep up. Go see the technology guy on Mondays, Mr. Horowitz. All right, so what's going to happen is one of the O's and the H are going to leave so that you can free up a bond, and then this is going to bond to this. That's what's going to happen over and over again, and what will come out is water. every time you bond it, you're going to come out with water. Is that clear? One of the H's, one of the OH's are going to leave, and what's going to come out is water, H2O. And that's why it's called dehydration synthesis. One of the H's and an OH is going to leave, and you're going to come out with water, H2O. And what you're going to get is a bond between oxygen and carbon, and that double bond sticking off to the side. Is that clear? Yeah. There'll be a straight bond. I'll have to draw it for you because I can't download it. So CH, right? This is still here. It's still an H. Now there's an oxygen, single bond, so it's going to be pretty straight. When the doctors are measuring your triglyceride level in your blood, they are measuring this. I'm going to put ... okay? How long is the fatty acid? Now, how long is it? It depends on what fatty acid got attached. Do you see that? The fatty acid, there could be many different fatty acids that would be here. They could be long ones or short ones. They could be saturated or unsaturated. Does that make sense? So that gives you variation. Which of these long chains you put on, that's up for debate. That all depends on what you're eating, how you process it, how much you exercise. And so what you're going to form is different kinds of lipids. Some of them will be high density lipids. That means that's bad. Wait, is high density bad or good? I always forget. All right. And then low density lipids, HDL and LDL. I can't remember. You'll have to look it up. So high density lipids and low density lipids, one's good for you. If you're going to need both in your body. You want one to be higher than the other. And I want to say, it's at this point of the day, I'm going to say I don't remember. But one of them is good for you having high and one low. Somebody, high density is good. That's what I wanted to say, but I wasn't sure. So high density is good, and you want this at a higher concentration. And low density is bad. But triglycerides are going to be, are they saturated? Are they unsaturated? All right. Are we all good with this? And why is it bad? Why is low density bad? Because they're generally going to be more saturated fats. The fatty acids are going to be saturated. They'll be straighter, so they're not going to be as packed together. Inside there's little transporter vesicles. You won't have as much in there. You'll have a lot more in one that has low, has double bonds, because you can fit more in the little ball of fat that's in your blood. And low density means you have more saturated fat, and they can layer and make plaques. And the plaques can take your blood vessel, a blood vessel that might look like this in a healthy young person that can fit red blood cells, no problem, through them. In an older person that has plaques, you might see something that looks more like, the plaques build up around the vessel. This is the blood vessel. And obviously the longer you live, the more you're eating like this, the less you get what's called blood clots happening, and you have a stroke, heart attack, embolism, whatever. They're all pretty much the same thing, stopping blood flow to a specific region of the tissue. A stroke is to the brain, Embolism is to the kidneys, lungs. Pulmonary embolism is to the lung, kidneys, liver. Right? Heart attack is pretty much an embolism to the heart. You're going to lose tissue. Muscle does not regrow. You kill muscle, it will not regrow. Is that clear? Just like brain cells, you cannot regrow muscle. So if you damage muscle, it won't regrow. Now, I said damage, not ripping muscle fibers, killing the muscle cell. There's a difference. Obviously, you want to tear muscle fibers so that you can stimulate more muscle fiber growth. But that doesn't mean you're killing the cells. If you kill the cells, they won't regrow. You kill skin cells, they'll regrow all day. Liver cells will regrow. You can take and transplant 1/3 of a liver, and you'll regrow that liver. That's not a problem. Kidneys will not regrow. All right. So we're all good with this, though? So that's lipids. Is this confusing for anyone? Does anyone have any issues with this? We can tell the difference between saturated and unsaturated. Okay. So that's pretty much it. Cholesterol is a ring structure. They form rings. There's various kinds of rings that are, and I'm just going to draw this really big ring, like it's a double ring actually. Cholesterol is a big ring structure, it's a double ring structure. There's various ring structures that are also lipids. What is a lipid? Mostly carbon and hydrogen. That's it. It's a hydrocarbon. All right. What are these used for? These lipids are used for this is all review, right? You read the chapter, this is all review, correct? Con review, we're all okay? Yeah. Long-term energy storage is generally the answer to that question. What elements? Now, are there other exceptions? Sure, they're also used for structure. For instance, phospholipids. Is the example, right? They're also used as hormones. Testosterone, estrogen are all lipids. Okay? So there are other uses, but the big use, most of the fat in your body, besides structural like phospholipids, is going to be long-term storage of energy. Is that okay? Do you understand that? Don't get tripped up. These do exist. Hormones, structure, so signaling is important as well. Nonpolar signals are important. Why are they used for signaling? Because they can pass through the membrane easily. They're nonpolar. Testosterone passes right through the membrane. Now, they're big, so they're not as easy as you might think, but it's a heck of a lot easier than sodium. Now, when you get to carbohydrates, what's their purpose? Their main purpose is what? Energy. This is what you call food. It's food. All right, so energy. Carbohydrates are energy. Short term generally, they could be long-term storage or medium-term storage. We call it medium-term storage in animals. And in plants of energy in the form of a polysaccharide. You agree? So glycogen gets stored in your liver for a short period of time until you go all night without eating because you're asleep and you need sugar. You're not going to wake up to eat a sandwich in the middle of the night every night. I know some of you do, but you don't have to. As you're sleeping, I know you do because my wife and I do sometimes. Obviously, I do. But you wake up, not a sandwich usually. And then you ask why I pick on him. So then, so when we're talking about medium-term storage, glycogen stores sugar in the liver. So when you don't have sugar, your brain can stay alive. Your brain needs sugar, so you need to have it stored away. Sugar you eat, you use. If you don't use it, you got to put it away somewhere. And that goes into a whole other signaling system we'll get into next week. But when we're talking about medium-term storage, you want to talk about polysaccharides like glycogen and starch. Starch is, the plants are saving their sugar. They're saving their sugar for their use. They're not making potatoes for us. They're going to use it to make a new potato plant. That's what they do. If you ever planted a potato, cut a potato, plant it. It'll grow a plant. It's ready to go. The eyes in the potato, those are embryos. They're ready to grow. All right, so that's medium-term storage. But there is one other use. Can you think of the other use? You should know, by the way. So energy, medium-term storage in the form of glycogen and starch. What's the other use, the third use? There's one polysaccharide that's missing here. What is it? And what's it used for? Structure, that's right. I love how she realized it. See, I told you should know it. You all knew it, you just forgot about it. It's cellulose. And that's structure. In plants, obviously. I hope nobody in here ever says cellulose is in animals. I hope no one ever says that. Cellulose is in plants. It creates the cell walls in plants. Are we good? So that's all the information. I'm going to give you a cheat sheet that kind of summarizes all this, but if you haven't walked through it, the cheat sheet's not going to mean anything. I hate calling it a cheat sheet. I think it's easy and fun, but it's not a cheat sheet. I don't want you. I actually had a parent argue that one time. She said, you called it a cheat sheet. Why are you mad that they used it to cheat?
She's not wrong though. She's not wrong. I don't like the word study, so.
I was like, it was a parent. But whatever. Assisted summary sheet. Let's call it a summary sheet. All right, so that takes us to lipids. That takes lipids and carbohydrates and their uses. Now let's look at, I should mention one other use that's important, but is often left off the list, identification. Carbohydrates are attached to lipids, they're attached to proteins to identify you. ABO blood group, they're usually glycolipids. We call them glycolipids and glycoproteins. Glycolipids end up identifying you. So carbohydrates by themselves don't, but if you combine them with when you start to combine these things, they can be used to identify you as being you. Why carbohydrates? Because they're so versatile. They can form all kinds of shapes. They can form long chains and then they can be branched. And because they can be branched, they can find all kinds of funky shapes that can make them specifically you. And you're the only you there is. Yeah. Go back. I think they've identified something like, I don't know what it is, like a bunch of different blood markers. I want to say 18 or 19 blood markers. Like we've talked about ABO blood groups, but there's actually like something like 18 of them, something like that. There, obviously you've heard of positive and negative, right? So there's ABO and then there's a bunch of minor ones, the positive and negative being the most famous, but there's all kinds of little ones. And each little one is another identifier, another surface, cell surface. Can I go on? Okay. Now, the last thing that I want to touch on today, I think we have time. Yeah, we have plenty of time. I'm not going to talk the whole period. I know.
Oh my God.
You have an essay to write.
I don't know
No. It's not really an essay. It's just I want you to. You'll see. It's an assignment. It'll be fun, ish. Ish. Better than listening to me talk all period.
I think we should talk. Yeah, talk.
All right. So protein. So the other kind of a big biomolecule, of course, nucleotides are going. Nucleic acids will be tomorrow. Proteins. Now, I will not be here on Thursday. I should tell you that. Somebody's mother is going to be here. She's a mother in the school. It's not Jonathan's mom. It's another student's parent.
Is it normal? I'm being for that.
There's 7 parents whose kids go to the school. You know what? It's something interesting. There are, well, of course, Sophia has parents, but they're not teachers. There's 7 teachers and principals can choose, and they can choose what school they want to send their kids to. Like we make enough money in this district that we can send our kids to private school if we wanted to. I think it's interesting that when teachers and parents and substitutes have that choice, they choose to send them here. Isn't that good? Doesn't that feel good? That people that you're sitting here, there's 7 of them that I know of that are in this school. More applied, but not all of them made it in. So I think that should make you feel good.
Are they in this?
No, they're a 9th grader. This particular one's a 9th grader's parent. Her sub will be here Thursday. Her mom will be here Thursday. Don't talk smack. Oh, I'm recording this, all right. Oops. Don't be disrespectful, and here's what I'm going to say to you. If you are not disrespectful and I don't get a negative report, then Thursday you're going to have a quiz on this material. Thursday's quiz will be open notes. All right, it's the only way I'm doing open notes this year is when I have a lot of doctor's appointments this year. If I'm absent and I don't get it. See, I'm not even asking for a rave review. I'm just asking not a negative comment.
Because you won't be here. You said you won't be here Thursday. But you said there's on Thursday.
It'll be online. It'll be online quizzes, open notes. Is that hard? You don't want it?
No.
It's like,
Want the open notes.
if you get the review after the Thursday, then because how would we get open notes if it requires this?
I'm doing it now. Aren't these the notes? This is what's
Do
the
you
quiz
think
going
the
to
quiz is
be.
on Friday or Wednesday? Because it would have to be on Friday since you're not going to be here. And then the sub is going to give you the review, and the day after we would have
Did I say the review? The quiz is on Thursday. I'm not here. You have the quiz online. AP Classroom.
On what occasion will we get open notes for your quiz?
That day, Thursday.
You said that if
You said if we're good, then we're gonna get.
if we're better with the sub, we get open notes. So
Yeah.
are you gonna have?
Oh, how would I regulate that? Is that what you're asking?
No.
If I get a negative response, we'll redo the quiz. I'll reset the questions, and it will be closed notes. I mean, does that not make sense? I just want you not to misbehave while I'm not here. That's all I'm asking. I don't expect you to. You're a great bunch of kids. Well, a couple of you I might expect to.
Is a student a boy or girl?
And if my chair's messed up, though, all bets are off. If my chair is not exactly how it is right now, as far as its settings, something's all bets are off. All right. So proteins. Proteins are polymers. These are true. Oh, by the way, lipids can never be polymers. Somebody asked why. You might ask, well, aren't these subunits? Couldn't you think of these as units that are being linked on? Yeah, you could. But the biggest one you're going to get is 3, right? Most of these lipids don't form long chains, polymers. Like these are units. Fatty acids can be thought of as units going on to glycerol. Yes. So you can think of these as monomers if you like. But we tend not to even discuss it in those terms. Does that make sense? We're not going to call a triglyceride a trimer of 3 fatty acids. You could, technically, but it's not, okay? And let's not even talk about it in those terms. Somebody might in the future, that's fine. But for the sake of your testing, to be clear, lipids don't make polymers. They don't make polymers. All the rest of these biomolecules do. So proteins are made of, are polymers. Polymers of what? Of amino acids. The monomer is amino acid. There's 20 different amino acids. You saw in your book, they give you a very good picture. It's an excellent, in fact, some of the older books, some of the more, I would consider the actual college textbooks that I have, that you guys, some of you took as alternatives, they're all older, but look, amino acids haven't changed. Their structures are the same. And the drawings are amazing. There's 20 different ones. Each one is going to be different. So there's 20 different. Do you have to memorize the 20 different? No. No one's expecting it. I can't. In college, yes. And if you're taking a biochemistry class, absolutely. Before you get into med school, you will have to know all 20 and how they react and which one reacts in what way under what conditions. Sure. But you're not a med student. You're going into college. You're finishing AP biology. This is an intro biology course. So no, you don't have to memorize the 20. But you just have to know that there are 20, we call these R groups. 20 possibilities. And the structure is actually a fun structure to draw. You have an amine group. That's one of those functional groups you should know. It's connected to a central carbon. I like to put little asterisks on it. You'll see an asterisk on a lot of the drawings. There's a hydrogen. There's an R group here sticking out. This could be any one of 20 of those R groups. One of those R groups is, wait for it, hydrogen. It's the smallest of the amino acids. And some are giant rings with long chains and rings, just really big. So big, small, polar, nonpolar, acidic, basic, ionized, or ionic, charged, rather. And then you have C, another carbon, and it's C double bond O, OH. And here's your carboxylic acid group again. You see it's the same group. Why do we keep saying the same structures over and over again? Because life is conserved. Let me pause here. So, but they're conserved. These structures are conserved. You'll see it over and over again. This is a carboxylic acid, carboxyl group. So this part acts as like an acid, and this part here is an amino. These are functional groups, so that together they're an amino acid. So this is an amino acid. How are we going to link them together? I think it should be pretty obvious by now that we're going to do dehydration synthesis. We've done it several times with several different biomolecules, and we'll do it again with DNA and RNA. So CH, there's your R group.
Do any of these biomolecules use hydrolysis, or do they always use?
All of them use hydrolysis. Hydrolysis is how we break them apart. Dehydration synthesis is how we build them. Good question. You use water to break things up, and you produce water when you build them. So when you work out with these muscles, you're working out, you break the peptide bonds. I'll define the peptide bond for you. You break the peptide bonds of the protein strands in your muscle. You're using water, you're putting water in. So your muscles will swell up. A lot of people get real proud. Look at my muscles. It's actually just water, most of it. And once you reform the new muscle, because you will add muscle mass, there will be muscle mass, built. You are building muscle, but you'll build a lot less mass than what you see initially. And once you initially do it, the mass will build, the bonds will reform. Water's going to come out. So you're going to go up in weight at first when you first work out, because you drink water to replace the water that you use to break the bonds. And then when you rebuild the muscle fibers longer, thicker, right? You're going to rebuild them. You're going to build muscle mass, but you're going to decrease in weight because you're going to expel all that water. You're going to urinate it out. So you gain and lose, gain and lose. Weighing yourself is the worst way of analyzing if you're gaining or losing muscle mass. At least weighing yourself at the moment, any one data point. The trend is okay. A trend line you can look at for weight, but don't look at weight to see if you're gaining fat or not. The best way to look at how lean muscle mass you have is your leather belt, not your spandex belt. Your jeans, not the spandex jeans, because spandex jeans do what? Stretch with you. Things that don't stretch, things that are standard, that aren't going to stretch easily, things that if they fit tighter, guess what? You're gaining mass. Maybe it's healthy, maybe it's not. Take a look at it, measure yourself. You cannot go by the scale. That scale means nothing. You could have grown taller. You could grow bone, muscle. Could have gained water weight. Lost water. I lost 4 pounds. It's all water. So weight goes up and down like crazy. I can show you weight charts if you like, but we're not going to do that. So just remember that moving forward. Your life is going to, too many people. Abs or water?
They're dressing as so tight, so.
Oh, you have a shirt with abs on it or something? Oh, I see. This whole type thing, it just confused me. When they first said it, type what? Are they typing something? What's going on? I'm like, oh, your generation, you have such fantastic words. It just takes me a while to learn them. Period. I did good, right?
No.
Okay.
Everybody, we're done. Class is finished.
Was that bad, huh? All right, so if we're connecting, if 1 is being connected to 2, what are we going to do? An OH and an H have to leave, correct? OH and H have to leave as water, H2O. And then what's going to happen, and this is going to happen over and over and over again. Yeah.
So we use hydrolysis to break the polymer, right? Is there any other way you can break it without using water?
You can, yeah, there's a ton of ways, but not that you're going to be able to live with. The way you live is dehydration, synthesis, and hydrolysis. If you want to do it in a test tube, you can burn it. You can increase the heat to boiling. You can increase it, put enough energy into it. You can break the bonds, but that much energy would kill you. So you don't want to do that. You want to control it with. So the energy required to do this is controlled with what? What are we going to use? What are you going to use?
What would you say? I don't. Yeah, not I'm stuck between. She's stuck between 2 abs.
This is going to be an interesting video to post. I wonder how many people are even going to understand what the heck you girls are talking about.
Stuck between 2 abs.
What are they saying? These people are crazy. No wonder public schools. I don't know. I'm going to keep going to private schools. I can't help this. So here's the rub. This is between 2 amino acids. This bond between two amino acids is known as a peptide bond.
Between two amino acids.
The bond between two amino acids is known as a peptide bond. If you know that, so this see that this is a functional group as well. The C double bond O in between these two amino acids. This is known as a peptide bond connecting to, and this is the linkage for amino acids. It's going to repeat itself. I'm not going to draw a bunch of these. That's ridiculous. I can't. I literally, emotionally, I cannot. So to repeat this over and over again, you literally do it like that. Some of these proteins, look at your hair. Look how long.
Well, you
Namada, stand up. Namada, stand up. Hey, think about this. Every tiny little section is probably what? 10,000 amino acids times from her root all the way down to the tip. And there's several individual protein strands wound around each other like rope wrapped around, threads wrapped around each other. That was all grown by your cells. How long did it take you to grow that? Like a year, 2 years, 3 years?
Not a year.
Do you keep trimming it?
Bald and she grows. I'm bald, so
I
there's.
trimmed it last year.
How low?
I think it was up to, oops, my abs.
Your abs fell off.
Like right here.
Okay, all right. So anyways, the point is that that's a tremendous amount of bonds, is my point. It's a tremendous amount of peptide bonds. That's why we call it a polypeptide. Because there's like, that hair has tens of billions of amino acids in a chain. By the way, if you have one R chain, if you have an R chain that has a sulfur in it, it's called cysteine. It's amino acid is called cysteine. If you have a lot of cysteines in your hair is real curly.
Oh, that's why.
If it has a medium amount of cysteines, it's wavy. If there's very few cysteines, it's straight.
Cysteine with a C or a S?
Look it up. It's a C. CY. Cysteine forms what's called disulfide bonds, and that's what I'm going to get to next, is their primary, secondary, and tertiary structures. We'll talk about that in just a second. So peptide bonds form between amino acids, and the R group is going to determine the property of that amino acid. What determines which amino acid gets connected to which in the order that it takes in? Write that question down. What determines the order of these amino acids? I'll answer it in a second, but you should know that. Peptide bonds, and so you can get a dipeptide. I hope you understand that means two peptide bonds, two amino acids. Or you can get a polypeptide.
Is it two amino acids or four? Because peptide is like a bond that connects.
That's a really good question. I love that you asked that. I had the same question when I first learned this. I was like, I'm confused. Dipeptide. Isn't the peptide bond peptide? So if there's a di, that means there's... Oh, I didn't say four. I thought three. So it's two peptide bonds between three amino acids. 3 amino acids. I said, no. They're just using it because there's a peptide bond in it, so they're calling it a peptide. Don't get me started. Not everything makes perfect sense.
So it only has one, actually, a peptide bond. But there's
Two possibilities. Either it did make sense and nobody told me, or, because that happens a lot, or I don't remember it, or there is no real good sense.
Wait, so one peptide bond, two amino acids? Or is it?
Yeah, one peptide bond, 2 amino acids. So that's 2 amino acids. So it's that's right. That's right. And dipeptide is 2 amino acids. It's a good question. Really good question. I'm really excited that you asked it. Very few people have asked it.
Very excited.
I really am. It means she's thinking. I can't tell you this whole year I've been so happy. You should ask Miss Sims. She was like, I'm glad you're having a good time because all my kids are actually, well, I shouldn't say all. All my kids are actually asking and participating, at least somewhat listening, at least occasionally listening. I've had years when it's been hard. Don't make me start crying. All right, so polypeptide, many amino acids. Many amino acids. So polypeptide, dipeptide. What do you think you might get as well besides these two? You might read about it, especially in your book. Oligopeptide. Right? In between. Again, something like 10. More than 10, more than 2, less than 10. So, anyways, these are polypeptides, and polypeptides are these chains. And again, they're these chains. At first, you're going to have 1, this is number 1, then it's going to get connected to 2, and then it's going to get connected to 3, and then it gets connected to 4, then it gets connected to 5. The little circles are the amino acids, the connections are the peptide bonds. And then whatever, right? Each one is going to have a different thing. This is R group 1, and this is R group 2, and 3. And maybe you get 2 3s in a row. Maybe you get 3 3s in a row. I don't know. And then a 4. Then you get another 1 again. Then maybe you get an R20. So each amino acid, the sequence is, there's a sequence. There's an order to them. And that order is going to define how this protein functions. I'll show you how in a minute. But first, the first question is, what determines the order of these? What order the amino acids are? What determines? You should know.
Is it what's in need? Yeah, like structure.
Speaking directly.
Whatever protein they have to make.
That's indirectly. That's what influences it. What determines, what says this is the way it's going to be? DNA. Yeah, I knew you knew it. It's just a matter of thinking about it. DNA has a gene. You know this. I'm just reviewing it. We'll get into details later. In DNA, there's a gene, and it continues afterwards. That gene codes for something. What does it code for? Directly. Directly codes for what? Well, specifically, that gene is going to code for what? No, codes, it makes what? What does a cell make from a gene? So the DNA gets turned into RNA. That's right. I know it's hard because I'm trying not to give away the answer. So RNA then does not have to, but can be turned into mRNA. And we will go into details about, this is where you're going to go into details about splicing. You're going to have to know splicing. Cutting things out. Exons get put together, introns get cut out, alternative splicing, all that. You're going to have to get an idea of what's going on there. RNA is going to go out of the nucleus into the endoplasmic reticulum. There's 2 possible paths to find what? Ribosome. Exactly right. Which is a big structure that acts as an enzyme and takes the message, the shorter message of processed RNA, and starts at AUG. AUG. And it continues down this. And yes, there's tRNA out here, that's correct. And each tRNA is going to carry an amino acid. It's going to have 3 anticodons. It's going to have UAC in this case. And that amino acid in this case would be methionine, so I might as well add it there. You know that, because as methionine is AUGs. I'm not going to get into translation right now, but the point is that this process of the order in the gene determines which amino acid goes when. You know that. Because that order is in the mRNA. Right? So this is what we're talking about when we're talking about order. Is it R1, then R2 and R3? Or is it R3, R3, R3? What is it? And that's going to determine, that order is going to determine these chemical functions. Because one R group might be acidic, another might be polar. So I'm going to use the simplest form to have this discussion, and I'm going to talk about polar and nonpolar, because that's the simplest discussion. You can talk about acids and bases, which is why putting a protein that's designed to work in your, like peptase, I don't know. There you have enzymes in your stomach to help break down proteins. They work in an acidic environment. Their shape is the shape they're in acid. Take them out of your stomach and put them in a basic solution, they lose their shape. They denature. They lose their shape. Because the acidity level, how many 8 hydrogen ions are available, are going to help determine the shape of that particular molecule. Specifically because the R groups are going to interact with the environment differently. It's not that the acid breaks the protein, the chain of amino acids, the peptide bonds are fine. You need peptase to break the peptide bond. You need an enzyme that's going to break those bonds to break them. So you don't really break it. When you say denaturing, you're not destroying the protein, at least not normally. What you're doing is unraveling it, losing its shape. So this is called the order of amino acids. This is going to be, I've seen this 100 times on AP exams. The order of amino acids is known as primary structure. This is the order. The order of the amino acid is primary structure. What order are the amino? It's the simplest of the structures. What is the order? Period. That comes from the DNA. The gene defines the order. How long is it, and which amino acid comes where? That's all. That's all they mean by primary structure. Are we all good? It's a sequence of amino acids in the chain. Is that making sense? All right. So secondary structure, who can tell me, who's done their reading, who can tell me with secondary structure? In fact, here's what we're going to do. You got 15 minutes left. I meant to end this a long time ago. I apologize. You guys wanted me to keep talking, but I didn't want to. I wanted to give you a project. But I'll give you 15 minutes to work together. Use your computers if you have to. You should be able just to use your notes. Ideally, since you've done your chapter reading, you've done notes on your chapter reading, and you have your summaries of your chapters that I gave you, you should be able to tell me what are secondary, tertiary, and quaternary structures. Go ahead and do it. Get together with your groups. Go across. Where's Abdul Bassett? He's still not here? Okay.